in the interest of time, we'll get going. I'm sure some more people will filter in as we start. So thanks everyone for coming along. This is the first talk this year of the Maths and the Life Sciences / Northwest seminar series in data science and mathematical biology. Our first speaker is Eder, who's a new Future Leaders Fellow at the university, started just in December from the University of Birmingham.
That's right,
yeah. And so he's just setting up his lab and his research. He's still recruiting some students, so I attached them to the advert as well. So please do circulate that as well.
Thank you.
And yeah, he's going to tell us all about his research in mathematical analysis of endocrine rhythms and what he hopes to do in his FLF here. So yes, over to you.
Thanks, Carol. And thank you and Igor and Odessa for organizing the seminar and inviting me. Well, as Carol already said, fairly new here to the system in Manchester, so still getting my bearings around and hoping that one day it will stop raining. Please help me with that. But anyway, I thought I would tell you a little bit about the research that we are doing. And I prepared my talk to an interdisciplinary audience because I understand it's not only mathematicians, but also some people from computer science and perhaps biology as well. But if there are any questions, just raise your hand. I'll do my best to answer them. So the research that I'm going to tell you about is very interdisciplinary. And as a reflection of that, this is the more important slide. So this is the acknowledgements for all the people that have contributed to these projects. Most of them are clinicians, clinical endocrinologists, but there are also some computer scientists and mathematicians, particularly Danny, Isabella, and Belinda, who's doing a PhD with me. And I guess the topic of my research could be framed within dynamical systems view of physiology. Perhaps you have heard about the word homeostasis, which is all these processes that try to maintain a fixed point or equilibrium in the body. But as I'm sure you know, nothing is fixed in the body. Everything is changing, fluctuating. So instead of a fixed point, we should be talking about a dynamic equilibrium, which is often described mathematically, perhaps a limit cycle solution or some other state of dynamic equilibrium. So as a matter of fact, all of this language that we use on rhythms and periods and amplitudes and phase, can be described mathematically. And it requires the coordination of several body clocks or oscillators in the body that must be acting in harmony. And this has led to new concepts in systems biology, such as chronodisruption, chronodiagnosis, and chronotherapies, which require the input of all these different disciplines. So we have emerging view of physiology that requires combining these different levels of expertise. And we have been applying this to one particular system which controls the secretion of cortisol. Now, cortisol is my favorite hormone because it helps us to counteract the negative effects of stressors. So contrary to popular belief, cortisol does not stress the body. It actually helps us against the negative effects of that stress. It can reduce inflammation, it can improve our cognition and memory, regulate our metabolism. But what's interesting to me as a mathematician is that it's fluctuating all the time. And if you measure it with high time resolution from blood, actually has this very interesting pattern of secretion. There are some ultradian pulses or less than 24-hour pulses that are modulated in amplitude in a circadian way. It usually peaks in the morning in humans, which is when we start our active phase, and then progressively decreases throughout the day. It reaches a minimum in the evening and then ramps up again while we're sleeping. The same pattern also happens in rodents, except that rodents, rats in particular, are nocturnal animals, so their active phase starts actually in the evening. There are both physiological and pathological differences in these rhythms in females and males, young and old animals, and also in animal models of disease. So these changes in the periodicity, the amplitude, and the characteristics of these rhythms matter. But the system that controls these dynamics is the HPA axis, or the hypothalamic-Pituitary-Adrenal axis. Essentially, the hypothalamus is this structure in the brain that receives all the circadian and stress inputs from the environment. and relays those signals via two neuropeptide hormones, CRH and ABP. The acronym doesn't matter, but essentially what they do is they travel via the microcirculation to the pituitary gland, which is not to scale. It's actually the size of a little green pea, and it's just right underneath the brain. And this gland is amazing because it basically decodes all the information from the brain down to this axis and other endocrine axis. And there is a small group of specialized cells there, the corticotrophs, which receive this information, and in response they produce ACTH, which then travels down the bloodstream to the adrenal glands. We have two of them, one on top of each kidney. And then the adrenal glands respond rapidly by secreting cortisol, which goes to all the tissues in the body. It can also cross the blood-brain barrier, which means that it can actually exert a feedback loop between the pituitary and the hypothalamus. And it's actually a negative feedback loop because it sends the signal to stop producing ACTH and these other hormones. So the dynamics of ACTH and cord looks like this in basal non-stress conditions. So this is if we are all happy and without any sort of stress. And as you can see, these dynamics are very nicely correlated with each other. So what is the cause of that pulsatility? And this is where the mathematicians made the first contributions to this system. It was hypothesized at the time that it could be the hypothalamus that was driving this ultradian pulsatility. But another possibility was that the feedback loop itself was the driver of these oscillations. So very simple mathematical model of three differential equations, actually a system of DDEs, because there is a delay here that is considering the time it takes for information to flow between the pituitary and adrenal gland. And these equations are representing ACTH, the glucocorticoid receptor in the pituitary, and the concentration of cortisol. And the prediction was essentially Hopf bifurcation was determining whether we will have pulsatility or not, depending on two key parameters in the model. So one was the strength of the CRH drive, which is the vertical axis that you see there. And the horizontal axis is the delay, which is the time it takes for information to flow between the pituitary and the adrenal. And essentially, the prediction means that there is like a Goldilocks region where if the adrenal delay is long enough and we have CRH values that are in sort of an intermediate region, then we will see the pulsatility. But if the CRH is either too low or too high, then we won't see that, which is essentially these simulations. All these are computer simulations. So the model was qualitative, it was actually quite simple. It was not fitted to data, but there was an experimental confirmation of the model 2 years later, where they took a series of, they took a group of rats and at the nadir of the circadian rhythm of corticosteroids, they just give them a constant infusion of CRH with different concentrations. And if the CRH was too low, as in the top left, then there was no oscillations of corticosterone. And as you progressively keep increasing the dose of CRH, then you'll see spontaneous pulsatility of corticosterone, then increases again, but it starts to become a bit noisier. And when you have very high values of CRH, then that oscillation disappears and you see just noise. And the column of the right, that's just the average of several animals. So all happy, we have now a model that explains this potential dynamics. But again, it's a model that is qualitative, it's not fit to data, and it's also for rodents, and it doesn't explain any sort of stress responses. It's just the basal scenario, which is what we are going to do next. So in normal physiological conditions, we have this dynamic equilibrium. So that is the homeostasis or homeodynamics, if you want. When we get stress, this could be physical stress. It could be that we hit our pinky with the edge of the bed and we shout, or we fall down the stairs, or it could be psychological stress as well. It could be that we get an infection. So there are many types of stressors that may temporarily disrupt this pattern of secretion. And this is a big problem in biomedicine, trying to understand how do these different stressors are going to disrupt these dynamics, and how do we return to the dynamic equilibrium? Why is it that our bodies can withstand some type of stressors but not others? And what if we are ill how is our body respond to a stressor if we already have an underlying condition. So this is very interesting from the mathematical point of view, I would argue, because if we manage to crack this problem, then we can support clinical decision making. So the stressors could be, as I say, physical, psychological, could be infections, could be environmental, or could be behavioral. All of these individual differences are going to play a role. So the timing of stressors is another one that has become particularly important in recent years. And just to illustrate this, I'm just going to show you a very interesting experiment that was done a few years ago. This is a classic experiment in neuroscience. So you have a rat that is living in the best life in a cage. It's the resident rat. And then you present a second rat, which is called the intruder. And several things could happen. It could be that the resident rat is friendly and starts, explore and becomes curious with the intruder. Could just that ignores the intruder rat, or could be that just goes off and attack the intruder, as these dramatic scenes show there. So what the experimenters did in this case was they took small volumes of blood from the tail of the resident rat, and later, a posteriori, they measured the concentrations of corticosterone. And these are the plots that you see at the top. So in some group of rats, the levels of corticosterone were going up, and in others were going down at the time at which the intruder was presented in the cage, which is this vertical rectangle that you see there at time 120 minutes. And then they took video recordings of the animals, and they quantified the percentage of time that the resident rat spent resting or exploring or socializing or attacking the intruder. And they just classified them according whether the levels of cortisol were going up or going down. And there was no difference in these three types of behaviors, except in the aggression behavior. So those animals that had the levels of cortisol going up at the time at which the intruder was presented became more likely to be more aggressive than those that the cortisols were going down. Yes?
Aggression is also include the fear. Fear is now lumped into response of the aggression, but there should be one thing that your aggression is introduced.
So I guess it will be difficult to quantify whether our aggression
is completely ignoring. So ignoring might be the case, right? You're hiding it. That would be cortisol inducing.
Well, it could be a response to the fear of the intruder. So it could be a bit of a I guess it's mixed with a fight or flight response in this case. But the only thing that you can quantify with the video is whether they are attacking each other or not. You cannot really quantify fear, but unless you do an experiment. It's a bit more. It's a different type of experiment for that. But I guess the message here was the likelihood that they will go in and attack the intruder does not depend on the absolute concentration of cortisol, but the rate of change, or the first order derivative, essentially. Yes? Yeah. It's not. It is, I'll show you some images later of how this is affected by your sleep patterns, your diet, and other sorts of behaviors. Potentially, yes, you're absolutely right. But they did a second experiment where they blocked the endogenous HPA axis by giving metirapone, which is a drug that blocks the feedback loop in the HPA axis. So it was basically a flat line. And then they give either metirapone with a vehicle or metirapone with a bolus of cord. And what they observe is those animals that receive the bolus of cord again show an increased aggressiveness versus those that didn't. So it's not just the specific phase of the day. Yeah.
So in this case,
Yes, the endogenous one is not present. Yeah. So essentially the message here is this sort of behavior matters in a way that we haven't quite understood yet. But in mathematics, we know that these are phase responses, and this is a behavior that was modeled a few years later, where they simulated cortisol and ACTH dynamics, which are these blue and red curves that you see there. And they simulated a stressor as a pulse of CRH. And if you give that pulse at different times of the day or of the cycle, then you'll see that the size of the responses in ACTH and core was quite sensitive to this particular timing effect. How does that relate to the aggressiveness? We don't know. We're just trying to characterize the dynamic behavior of the system. Right. Let's look at another example in humans. Heart surgery. Coronary artery bypass graft is a very common type of intervention, has a high survival rate. And I know it looks very traumatic there in that diagram, but most people are discharged the day after and they just go home. But a small fraction of individuals have an excessive response to the surgery, excessive inflammatory response, and that increases the cost of hospitalization, and obviously they can sleep and they feel quite unwell. So we partnered with a group of anesthesiologists and cardiologists in Bristol to look at data from 10 individuals that went through this heart surgery. The demographics was quite uniform in terms of their, BMI, the duration of the surgery. It was all done in the morning, maybe plus minus a few hours, but not much really. And we wanted to see how do the ACTH and cortisol will respond to this type of stressor. At the top right, this is just an example of how a normal 24-hour profile will look like in humans. And at the bottom is where one example of one participant that had heart surgery. So there was an increase in both the baseline of ACTH and cortisol, but also the periodicity and the amplitude of those ultradian pulses was larger. Now, this is just one example. Oh, thank you. And what Danny was doing is first start with a data analysis approach. Try to characterize the synchronicity between these two signals, just calculate the ratios between these hormones. The set score is to be able to map them out in units of the standard deviation, calculate cross correlations, standard statistical techniques, can quantify the periodicity, autocorrelations to look at the lag between ACTH and the cortisol signal. This one is particularly interesting. This is a rolling window time lag cross correlation. So basically calculate the cross correlation at different epochs throughout the entire time series. So in each horizontal slice is one epoch and time flows downwards. So we start at 0 at the top and then we go downwards all the way to the last epoch. And if you see a very organized and regular pattern of vertical stripes, that means that ACTH and cord are synchronized with each other across the entire time series. And if you see those stripes like shifting a little bit, that means that they start to desynchronize at some point. So all this is very regular for the healthy scenario. And this is how it looked like for the 10 participants. So panel A is just the control. Panel B, we found 4 participants that had at least 2 large pulses of ACTH and cortisol. Panel C, these are individuals that maintain some level of ultradian pulsatility, have their baseline increasing. And panel D, they just had just one massive excursion of ACTH and cortisol with no rhythmicity at all. So it's a bit of a way that we try to discriminate between these different types of phenotypic responses or dynamic responses to the stressor. But what we did then was to develop a sort of trying to understand this phenomenon based on the previous knowledge that we had about this Hoff bifurcation model of the stress response. So what we think is happening is there is in baseline conditions, the system is entering and exiting this pulsatile response region every 24 hours. And a stressor may temporarily disrupt the system away from that dynamic equilibrium before it returns. So the idea at the time was maybe what is happening is that the way that the system responds to the stressor depends on how it is perturbed within this region. But that was entirely qualitative, so we didn't have a model at the time. We did a mathematical model, but it was a parsimonious model, just basically an open loop system that was calibrated to the data, and we tried to identify by fitting the model parameters to the data, which were the mechanisms that were more likely to be involved in this sort of distinct phenotypic responses. So all of this is published, but essentially what we found was that the two of the phenotypes, 2 pulse response and the ultradian pulse response, was solely explained by the model. But the 3 participants that had this massive excursion of ACTH and cortisol, these dynamics could not be explained solely by the model. So we hypothesized that there must be something else that was intervening there, perhaps inflammatory mediators that were also measured, but we didn't develop a model for that. That's still pending. So if anyone is interested and has experience in model identification, we have lots of data from inflammatory mediators to do that. And we still have to see whether that cohort matches with those individuals that stay longer in the hospital after the surgery. It's quite difficult to get data from these patients, to be honest. Right. So this made us realize that we had to update our models, our understanding of the HPA axis. So Belinda started her PhD 2 years ago in Birmingham, and she has been having access to data from healthy humans and from patients, and we're using that data to update our knowledge of the HPA axis. So first of all, she did a more systematic analysis of pulsatility using wavelets. And this wavelet analysis allows us to quantify that periodicity. She developed a delayed differential equation model of these pituitary-adrenal feedback loops. Now the model is calibrated to human data using a parameter inference for noise time series, which is HINDS, this fantastic piece of software developed by Gary Mirams et al. In Nottingham. And the predictions of the model are this one. So this is just the cohort model that predicts the ultradian pulsatility in ACTH and CORT. What is important of this model is now the dynamic range for ACTH and CORT is what is actually observed in humans. So it's a quantitative model. The pulses have a period of about 2.5 hours, which is what is observed in humans. And there is an asymmetric circadian drive, which is different than in rodents. So the rodents have 12 hours phase going up and then 12 hours going down, whereas in humans, we only have 8 hours going up while we're sleeping at night and then 16 hours going down. So it's a different pattern of secretion. She was able to reproduce the experiment that we saw before, where we have the high, medium, and low levels of CRH. And this is model prediction from using DD tool, this toolbox in MATLAB to do bifurcation analysis for DDs, where we again see the distribution of delay values falling squarely within the ultradian pulsatility region and the low, medium, and high CRH stimulation. Now, what are we using this model for? Well, we thought, well, let's see if we can reproduce this qualitative behavior that we observe in the patients that had the heart surgery. So some of them have just one big pulse of ACTH and CORT, others have 2 pulses, and others have just multiple pulses. And she used the model to simulate how stressors of different magnitude, duration, and timing could affect the observed dynamics that we see in the system. In some patients, surgery lasted a little bit longer than in others, or occurred 1 hour later than in others. So we don't know how is that affecting these dynamics. So essentially, she is using this model as a test ground to see how all of these different characteristics in the stressor may affect the dynamic behavior that is predicted. And this is just a summary of what we think is happening in the system. So we have levels of CRH that go up overnight every 8 hours, and then they go down to 16 hours. And then we have these perturbations or the stressors that may deviate the system from the dynamic equilibrium before they are brought back by the homeostatic regulation. And that's just considering the pituitary-adrenal feedback loop. We haven't incorporated the hypothalamic feedback yet into the model. So that's something that we still have to do. The next step she's doing, oh, by the way, that was submitted last week to publication, so fingers crossed. But the second paper for her PhD is to look at the effect of sleep disruption. So we got data from this paper where they took a cohort of 23 healthy individuals and they were volunteering very bravely to a 48-hour continuous wakefulness protocol. And every 2 hours, they gave a small sample of blood. And from that blood sample, we measure ACTH, cortisol, and other adrenal steroids. And essentially, the average data looks like this, where here in blue and in red, you see the controls. And in yellow, in each case, you see the sleep-deprived cohort. So as you can see, cortisol seems to be very similar to the controls, but ACTH seems to be quite out of phase. But when we look at the individual data, we see the same thing that we saw for the heart surgery case. Some individuals seem to be more vulnerable to disrupting their hormonal rhythms due to the sleep disruption than others. So there is some sort of phenotypic heterogeneity there in the response to the stressor that we're still trying to characterize. So what she has been doing is developing. Yes.
Can this be someday a biomarker early on? Is there any cohort, much larger study in STS?
For continuous wakefulness, no, there isn't. This is the first one that we are aware of that measures ACTH in cohort. Well, there is. Sorry, no, I'm lying. There is some data, but it's not at high time resolution as this one. And there is some studies from the 1990s. I'll show you some figures of simulated shift work and phase synchrony protocols, where you see cortisol also changing in their circadian phase with respect to the controls. But ACTH had not been measured before.
Blood samples in 24 hours. Do you take
Sorry?
12 blood samples in 24 hours?
Yes, it's a lot. So for the healthy controls, for example, 24 hours, you cannot do it for longer because the amount of blood that you give is equivalent to a blood donation. So you need to stop it after 24 hours. And I actually volunteer for that study. It's very uncomfortable because you have to be there in the hospital. Anyway, so the question is, how do we characterize this misalignment? And she has been calculating things like area under the cure, the peak, the concentration at the nadir, the time at which the peak and nadir occurs, autocorrelations, and other metrics to distinguish the control data from the sleep-deprived data. And input this into a machine learning classifier. This is still work in progress, so these are very recent graphs, so that's why they look a bit, they need some polishing. And what she's doing now is to couple the model of the HPA axis with a two-process, with a mathematical model of sleep, which is known as the two-process model, where you basically assume that there are two competing pressures, one that tries to wake you up and another one that tries to put you to sleep. And that mathematical model is now being used to predict how this sleep disruption is going to affect the stress hormone dynamics. And we also want to use it in the future to test how stressors can in turn affect your sleep-wake processes.
But there must be some field recognition data and you can find it.
I mean, I'm sure there is.
And any maybe much larger cohort where you can.
It's difficult to like you ask people,
Some people
have they, to characterize it by this. I think.
So a few issues are, well, for example, whether the data is self-reported or was it measured directly by some sort of wearable device. I'll show you something in a moment. But also time series data is like gold dust for mathematicians because we can then fit a model to that time series. So that's why we just try to get our hands on this data set first. Yes.
Does the cortisol levels in the control population sort of look like what you expect? Because again, I imagine it's quite stressful having 12 blood tests at 24 hours. That like generally cortisol level, right?
Yeah, that's a good point. So yeah, in fact, as a matter of fact, yes, having the blood collected there and just being there can be stressful in itself. So we usually discard the first time points to allow the system to kind of thermalize, that we say in physics, or reach its equilibrium. But Yeah, that's one of the things that you cannot really.
The
There is it's a cannula that is permanently attached to your yes, and when you're trying to sleep, you cannot roll over, so you have to stay like, almost sleep like a vampire, Right. Now, this conversation is ideal for what follows next, because measuring cortisol in different compartments is quite challenging. So what most people do is measure from saliva or from urine, because it's easily accessible and it's cheap, and most labs around the world can detect these steroids from these compartments. The problem is that they are excretory fluids, and what they contain there is the cumulative dynamics of several hours prior. And you can also measure it from tears or from hair. And I'm sure there's a joke to be told there about how stress can be measured from tears. But so the gold standard is blood plasma, but as we just commented about, is quite difficult to measure, and it's expensive and uncomfortable for both the participant and the clinician doing the collections. You have to stay overnight just collecting the blood. So what we're trying to do now is collect from interstitial fluid, which is the fluid that permeates the space between our cells, basically. And our colleagues have developed this fantastic piece of technology. It's a wearable device that you wear around your waist. And sorry about the gory images, but essentially, this is a catheter that goes underneath the skin. The catheter is a flexible plastic tubing about 0.5 millimeter in diameter. And on one end of the tubing, you have a pump that is pumping saline solution every 20 minutes. It pumps a column of fluid, column of air, saline air, and so on. And the section that goes in the skin has a semi-permeable membrane that allows the passive diffusion of small molecules into the saline, but not the other way around. And then at the other end of the catheter, you have this device, which is a collector. Then you can take the spool, and each of the columns of saline corresponds to a time point, and then you can reconstruct the dynamic profile of secretion of these hormones. And you can wear this device for up to 3 days continuously. You have one time point every 20 minutes, and you can use it in real life settings. So you don't have to stay in the hospital. You can go to work, you can sleep with it. So you can measure these dynamics in real life settings. Sorry?
why
It goes into adipose tissue, yes. So subcutaneous. Sorry? Subcutaneous, yes, it's fat tissue. In principle, you can put the catheter in other parts of the body, but we have only tried in the abdomen. It's another diagram how it looks like. So basically, subcutaneously, you have the fat tissue there. So the corticosteroid profiles, there's quite a lot of variability in them, and that's one of the challenges that we have to overcome, how to quantify it. So in the study that you sent, was funded by EU grant, so that sample over 500 participants, 214 healthy volunteers, and the other had fractions of people that have several endocrine conditions. This is the demographics for the healthy cohort, and this is the number of adrenal steroids that were recovered. So it's not just cortisol, but also cortisone, aldosterone, which is important in the regulation of blood pressure, corticosterone, and other metabolites. This is an example of how the data looks like. So this is 214 profiles from healthy people. So each of these blue lines, projector is not very good, but you squint your eyes, you can believe that it's in. Or you can look at the reference, so you can see the high resolution of this image. And so we calculated the statistical moments, and essentially we have data from all of these steroids. So this is the steroidogenic pathway. So cholesterol, which is the precursor for cortisol, will be at the top, and then enzymes are going to produce all of these different hormones, including cortisol, cortisone, and aldosterone. Some of them have a clear circadian rhythmicity, others are less obvious. Some of them have a lot of variability, others less than others. So we have the challenge then of quantifying that variability and do something useful with that. So we did the basic stuff, first calculate some patterns of secretion, use a rhythmometry analysis, cosigner analysis to identify those ultradian pulses of secretion. look at the phase differences between the hormones. So this circular heat map is like a 24-hour clock. So you have midnight at the top and midday around the bottom. And each concentric ring is the 24-hour average of all the participants for each of the different hormones that we saw. The white arcs are the time at which they went to bed, and the fall asleep, and the black ones are when they wake up in the morning. And we have data also for the meals, breakfast, lunch, dinner, etc., and so on. So what we observe actually is that those hormones that are upstream in the steroidogenic pathway, they tend to peak earlier than those that come downstream. So that is a reflection of how long it takes for the hormone to be synthesized and the signaling cascade to pass down. And there is a PhD visiting student from Germany who was doing a mathematical model of this pathway, and that still needs to be written in the manuscript. The biomarkers. So the clinicians were interested in how can we use this type of data to improve diagnosis. So again, very simple things. Let's calculate these biomarkers, the area under the curve over different time windows, even nadir, morning peak, and so on, and calculate the distribution of those biomarkers for the cohort. So these are what these violin plots are showing here. So this is the distribution of these values. And the horizontal red lines is the value of those biomarkers for one candidate profile. So we can compare against that distribution and see how normal is that individual compared to those biomarkers. And we have been using that for the patients. So in the left column, we have the example of one patient that have a disease called Cushing's, which is excess production of cortisol. And what we found is it's not just excess cortisol. There's actually an abnormal pattern of secretion of cortisol. So that red line that you see at the top is an ultradian pulse of cortisol, but that's a very high amplitude compared to the background of the healthy, which is the shaded blue area that you see there. So some biomarkers seem to be within the normal range, but others are clearly above, as you can see in those plots over there. Another condition was primary aldosteronism, which is excess production of aldosterone, and is the second largest cause of hypertension. And again, abnormal patterns of secretion of aldosterone, and the biomarkers telling us information about how abnormal is that pattern. Isabella has been instrumental in taking these biomarkers, putting them into a machine learning classifier, and identify which features are the most important to discriminate between what is normal and what is abnormal. Because we are aware that this technology, as impressive as it might be, is not a one-size-fits-all. So you cannot give this device to every single patient. So, but the question is, what hormones you need to measure and at what times of the day, so you can give a confident prediction of whether something is normal or not. So that's what she has been doing. And another project that we had is to look at patients that have adrenal insufficiency. So in these patients, the adrenal glands are not working properly, so they don't produce cortisol. So they need to take tablets of hydrocortisone that try to restore the normal circadian rhythm. And this intervention hasn't changed since the 1950s, 60s. So you take one big tablet in the morning when you wake up, and then small dose during lunch, and then a smaller dose during dinner to try to mimic that circadian pattern. And it keeps the patients alive, but they don't have the same quality of life that a healthy person because it's not restoring the physiological pattern. So this is an example of one of those individuals. So you see big dose in the morning, and you see a spike of cortisol, which doesn't quite look like the physiological average, and smaller doses with the dinner as well. So some patients have 3 doses, others have 4. Formulations change. It's all very artisanal. So what if we could use these mathematical models and these biomarkers to optimize this hormonal replacement therapy, to tell precisely how much of the dose you need to give and at what time of the day to normalize that rhythmicity. So that's another project still in the works. So someone interested in PK/PD modeling, please drop me a line. Now, what do we want to do next? And this is part of the fellowship work, is how we can use mathematics, computer science to develop this new type of tools for preventing, diagnosis, and managing endocrine conditions. So it's all about having some actionable clinical insights. And the best example I can think of to show this is shift work, because it's a type of disruption that touches upon several sources of misalignment. So a shift worker is not just someone that needs to stay awake at night. They also have to eat at that time. So they eat at the regular hours, and they are exposed to artificial bright light because they will work in a hospital, or so it's not natural light conditions. And they are often first responders, which means that they are experiencing stress during that time. So it's a very complex environment. And well, shift workers are at a much higher risk of developing endocrine conditions, cancer, and other diseases. They have a shorter lifespan. And it's about 20% of the world population that does some type of shift work. So it's quite a lot of people affected. UKRI funding landscape is changing, and now they want to solve problems like for yesterday. So using wearable devices is a good option, because it will allow us to move care from the clinic to the hospital or from at-home environments. But we need the maths, we need the theory to connect the internal dynamic landscape that we see in the body for health to these non-invasive wearable signals. So someone was asking me before about whether glucocorticoids have been measured in shift work. Yes, there's examples of this. So in active individuals, melatonin, which is a hormone involved in the regulation of the sleep cycle, increases overnight, peaks, and then goes down before we wake up, whereas cortisol looks like this. And in studies where they have done for the synchronic protocols, there is a misalignment of cortisol, but also other hormones involved in the regulation of metabolism. This is an example of this participant on the top. The black line is when they are sleeping, and BLDS is breakfast, lunch, dinner, super. So the first 2 days, 3 days is all normal. And then in the following week, they're progressively asked to stay, to go to bed a little bit later and to shift their meals later and later and so on. And we see that misalignment affects the phase shift and the amplitude and the dynamic response of these endocrine systems. So essentially, instead of having a synchronicity of hormonal rhythms, what we have is a choreography, because not all the hormones are oscillating at the same phase or at the same time or at the same amplitude. So how is all this maintained? And how does a chrono disruption is going to affect the amplitude and the dynamic response to a perturbation? So that's a big question. And this is where I think that mathematical models can fill those knowledge gaps. So imagine that we have the cortisol oscillator and the melatonin oscillator and the glucose responses. They are all exchanging information with each other. Endocrinologists, they often investigate the systems in isolation, but now we know that they are exchanging information, and they are subject to external perturbations. Could be the stressors, could be sleep disruptions, or mistiming of meals. So how can we shed light on this? This is where the wearables come useful. So we have done a pilot study where in 11 participants, they were fitted with the Eurorhythm device. And at the same time, we gave them the Aura ring to measure heart rate variability, accelerometer, body temperature, finger temperature, Freestyle Libre continuous glucose meter that you put in the shoulder to measure glucose, the motion watch also measures light exposure. You look like a bionic person with all your wearables. And we measured them for 2 weeks continuously. And this is just some examples of the data for cortisol. Each ring is 1 participant, so average for 24 hours. Melatonin, so we can measure it continuously as well. So every 20 minutes, they on glucose and so on. And one of the questions that we want to answer here is, can we infer this internal hormonal milieu from these non-invasive signals that we can collect with wearables? And in the middle, we have a black box that we don't really know how it works. And this is a problem for mathematicians. And we know that stressors can affect your heart rate variability and maybe physical activity. And your blood pressure can relate to levels of aldosterone change in your posture, whether you're sitting down or you're walking or you're laying horizontal. So there is a lot of model inference to be done there. And I'm trying to find what works really. So please help. And this is one of the potential applications that we want to develop. I'm almost done. This is a pilot project that we started 2 years ago, but then we ran out of funding. So essentially what we want to do is, if we know that these devices like the urethrum are quite expensive, it's an enabling technology, but what the future is telling us is that we are going to have at some point sensors that allow us to measure these hormones from patches, these skin patches or subdermal implants that can give you a signal of how much cortisol, how much stress is in the body. And we could get ahead of the curve and try to develop the theory to interpret the signals and give it useful information for the user, would be the patient or the clinician. So similar to what the artificial pancreas have done for patients with type 1 diabetes, where you have your glucose meter, sends a signal to your phone, it tells you whether your glucose is too high or too low, and then you can send a signal to your insulin infusion pump to give a certain amount of insulin or a period of time to restore that normal rhythm. So we could do the same for cortisol, perhaps. And in order to do that, we need to develop the maths. We need to develop a mathematical model that explains what should be the target range for cortisol at what time of the day, and how much should be secreted by the pump in response to what patch is reading at the time. So that's the dream, and we have some projects with Imperial at the moment to develop that patch, but I don't know much about it. I'm not biochemists or microelectronic experts, but just trying to focus on the models for now. And this is my last slide. So hopefully I've convinced you that we need to shift from a static to a dynamic view of endocrinology. And endocrinology is becoming more of a quantitative science, as is all of medicine. I personally feel that it's in the same stages that neuroscience was maybe 10 years ago. And now we have a lot of computational neuroscience departments in universities. There are so many open questions to be addressed here. And trying to understand hormonal rhythms is going to require this time series data, but now it's available. Now for the first time we can measure it. And also how does it respond to perturbations? The digital biomarkers have been quite useful to help some patients to adjust their doses in hormonal replacement therapy or accelerate diagnosis. It's not policy yet, but it has been used. And we need to develop this theoretical understanding of how these endocrine systems are talking to each other. There is a quote by Sidney Brenner I really like because it summarizes what you have seen here. So it says, progress in science depends on new technologies, new discoveries, and new ideas, and usually in that order. So we have new technologies to measure these hormonal rhythms, and we have new mathematical and computational techniques to make sense of that data. And that has led to new discoveries about variability and about how the system responds to these stressors. But what are the open questions or the new ideas? So there are a few of them. Pick your favorite. Why are hormonal rhythms so common instead of just constitutive secretion? So why not produce a hormone all the time? And there are two hypotheses. One of them is the energy hypothesis, because it's cheaper for the body to produce a hormone impulses than having the switch on all the time. But there is also the information hypothesis, that you can pack more information into an oscillation in the amplitudes, in the periodicity, and in the shape of that secretion pattern, which leads to the second question, which is how is that information decoded by the target tissues? So you measure a hormone from blood and you see these rhythms, ultradian and circadian, and all the body sees those same rhythms. They respond differently. The liver will do one thing in response and the pancreas will do another in the brain. And that has to do with the receptors that are interpreting that signal and how they are distributed differently in those target tissues. So there are a few projects that are quite interesting to look at. What are the sources of variability? So intra-individual variability and inter-individual variability. So how does these rhythms look like in people from different sex, age, BMI, but also their chronotype? Morning people or morning larks or night owls are going to have a different pattern. Or if you live in Scandinavia, where you have different duration of day and night, it's going to be different than we see that in our participant sample in Norway versus those sample in Greece. Their diet is also different. How does that affect variability? Well, we don't know. And this is my favorite one. What makes these endocrine rhythms robust to some perturbations, but fragile to others? So you can hit your pinky with the edge of the bed or skip a meal, and you're probably going to be fine the next day. But if you always eat at irregular hours, or if you are experiencing some sort of chronic stress or severe stress, that is going to misalign your hormonal rhythms and make you more vulnerable to other conditions. So what is the threshold? How much is too much or how long is too long? And I think that's a problem that we can also address with these soft models. And if you have a perturbation in one endocrine system, how does it cascade down to another? And well, can we use all of this insight to design strategies to realign these hormonal rhythms and restore health? Well, I hope the answer is yes, but that's all I got. Thank you so much.